Anne Stone to give one of our first lectures of the fall. So Anne and I have known each other for many years. She was a senior graduate student at Penn State University when I began my PhD studies, and she was the example of the perfect graduate student because she always had things organized, at least to all of the rest of us. Everything always seemed under control. She says that all of the anxiety was hidden, and I think that's because all of her anxiety was hidden in a closet on the top floor of the building, where she spent most of her time innovating ancient DNA studies. So she was at the very beginning of this incredible technique, this incredible new type of science. And as you'll see in this talk, she's going to tell you really where it's gone. So she has been from the birth of its creation to the pinnacle now of how these studies are being used to improve our understanding about human health and how our biologies came to be the way they are today. So please help me welcome Anne to the stage.
Thank you very much. It's a real pleasure to be here. Hopefully I'm loud enough. Sometimes I speak a little softly. So yes, I want to talk to you a bit about how we study ancient pathogens, actually, and a little bit about the story of ancient DNA generally. A lot of times people say, okay, you're an anthropologist, why disease? Why are we so interested in pathogens? And I think there's a number of reasons. One is just in recent history, we know they've had a major impact. We've all just been through a pandemic, of course. But when we look at our genome, the biggest signal of selection is always pathogens. It's really the immune loci that are the first to light up on any genome scan that we do of any modern population and even any ancient population. It's really the immune loci that light up. So we know that has been a consistent pressure over time. You know, when I give a talk generally to the public, I also emphasize that really understanding the evolutionary history of ourselves and of pathogens helps us think more about designing better vaccines and drugs in the future. So even though what I do is very basic research, it helps us think about how these pathogens evolve, how we can design things better to combat them, because as we have learned most recently through the COVID-19 pandemic, you know, pandemics are not something that we've done away with, right? There's always going to be another one, I'm afraid. Now, really from the start of ancient DNA research, people were interested in pathogens, but what we could do was pretty limited, right? And so today, one of the pathogens I'm going to talk about is Mycobacterium tuberculosis, which is actually part of a group that causes the disease tuberculosis. And the first study of Mycobacterium tuberculosis was in 1994. It was published in PNAS, and this was their fabulous result. A band. And basically, and this is a repeat element. So sequencing, they did sequence it, and it showed that it was the repeat element, and it's a repeat element that's found in the Mycobacterium tuberculosis complex. So Mycobacterium tuberculosis and related pathogens, and it basically said, yes, this mummy that has these signs of TB has TB. Ta-da! But it was, you know, a tour de force at the time. And I like to point out how technology has changed. This was back in what I call the floppy disk era, the smaller floppy disk era. And those of you who are much younger are probably like, what? And then we had these hard kind of 3-inch discs, and of course now we don't use them at all. And it was also in the time of Sanger sequencing, so this was really laborious. We were using PCR, we were doing this laborious type of sequencing that could only really give us if it wasn't ancient DNA, the longest reads were maybe 1,000 base pairs. Ancient DNA is typically much smaller, as you'll see. So the revolution in ancient DNA has really gone in tandem with the technological revolution and a bioinformatic revolution. And so methods have changed a lot. So how do we analyze ancient pathogen DNA? So at ASU, we have a clean room. It's a class 10,000 HEPA filtered room. So during a pandemic, it turns out to be the safest place to be because all the air going in is HEPA filtered down to the level of viruses. So we were able to keep working. But what we do is we take a very, interesting. Imagine a bone cartoon in that spot. Oh, this will be fun. Right, so you take your sample, which is very small, but usually visible, and we can use about 50 milligrams, right? So very small. Initially, we started with more, but it turns out smaller amounts are better. The amount of DNA, though, that we get from an ancient bone or tooth or hair or whatever is usually, though, less than 1% is endogenous, right? So most of the DNA that we recover is from the environment, the burial environment. And that, as you'll see, makes things challenging. We then take the DNA and we make a library. And this means we take all that DNA from all those sources and we add these adapters. We then add little barcodes or indices. And more adapters, and this really lets us manhandle the DNA or work with it and grab it in different ways. And okay, so we're not interested in all that DNA. If we sequenced it, most of what we sequenced would be just from the environment. A lot of it we wouldn't actually know what it was from. So instead what we do is we go fishing, and we use baits. These baits have little beads on them, streptavidin beads. They we use basically chemistry, yay, to add magnetic beads. So it's all biotinylated, and then we can use a magnet to pull this to the side and pull out and enrich for the DNA we're interested in. So if you're interested in human DNA, you use human baits. If you're interested in a pathogen, you design your baits toward that pathogen. And then you remove the rest of the DNA, and this enriched DNA is what you sequence and then analyze on your, using your bioinformatic pipeline on your cluster. So, this methodology really came out of the Neanderthal Genome Project, to be honest. A lot of it was very much developed and improved during that process. So, okay, how is dealing with ancient DNA tricky? You've all kind of, I'm sure, heard that it's hard, and it is. There's a couple of reasons. One is the extraction methods that we use to pull the DNA out. Typically, prior to newer methods, everything that was less than about 70 base pairs was lost. Now, the thing you'll notice here is that if you do just shotgun sequencing, so you just sequence all that raw DNA, your peak is at about sometimes 30 base pairs. Mitochondrial DNA capture, okay, you'll notice that the peak is somewhere in the 30 to 40 base pairs, right? So you were losing most of your DNA prior to improvements in the extraction methods. The other real challenge is that trying to analyze the data bioinformatically, you really can't analyze fragments that are smaller than about 25 to 30 base pairs because they'll map to multiple places across the genome and you won't know which is the right place, okay? So what you'll notice is there's kind of this sweet spot of where we get most of our ancient DNA data. And so this is another hard challenge. The other real challenge is DNA damage. So after death, your enzymes aren't working hard to keep your DNA in tip-top shape. And what happens is the DNA starts breaking down in some interesting ways. But one of the things that happens is you get these sort of ends that are single-stranded, and that's where you tend to see these deamination is the type of damage that's most common. And when you do that and you get your sequences, what you notice is that a cytosine, when it deaminates, basically looks like uracil, and that's read by the sequencer as a T, a thymine, rather than a C, okay? And so it looks like you have all these C to T changes at the ends. And so bioinformatically, we have to adjust for that. So that's the other big challenge we face with ancient DNA. Okay, so, but what about getting a pathogen? What are the challenges there? And there are quite a few. Most bacteria and viruses are not known to science. So that's why I say there's this kind of dark matter of the genomic universe of things that we don't know what it is, okay? And if you think about pandemics in the past, some of them, you know, historians have a pretty good guess because the descriptions were pretty good about what that pathogen was. But there's a lot where we have no idea, right? And so one example, in the Tudor period in England, there was this outbreak of something called the sweating sickness, right? There's a symptom, an ill-defined symptom for you. And, you know, it affected a bunch of people. We have no idea what it was. It's not described later. And so, you know, we would have to find somebody where we knew they died of this sweating sickness, and then we would have to hope that there was a pathogen load sufficient for us to figure it out, and it was related to some other pathogen we know about, right? We also have the problem of a lot of pathogens have very closely related environmental relatives, particularly Mycobacterium tuberculosis. There's a lot of mycobacteria that live in soil and water. So how do we find these needles in the haystack of DNA that we're looking for? So this is actually from some of our earlier work where we are trying to pull out the DNA and identify the pathogen. And one of the things we learned was that distinguishing between the mycobacteria that were in the TB complex and distinguishing between the environmental mycobacteria is often very difficult. And that's one of the major hurdles when it comes to, it's why you don't have as many ancient DNA papers for TB compared to some of the other pathogens, because there's a lot of environmental mycobacteria that we have to pull away from it. Another real challenge that we have when it comes to pathogens is bacteria and viruses do some crazy things in their biology and in their history. They recombine, they exchange genes, they have plasmids, they have some crazy substitution rates that have a lot to do with population dynamics. We've seen that with the SARS-CoV-2 pandemic, where when there's this huge outbreak, you can actually get what looks like to be a real increase in mutation rates, for example. And so we have to figure that into our models, and that can be quite challenging. There's some pathogens like the one that causes gonorrhea, which is a sexually transmitted infection, that exchanges genes like crazy, and it's an awful thing to try to study from a population genetic perspective, because you have to treat it almost like it's a pan-genome. So you don't see ancient DNA studies on that because it's so challenging. The other thing we have to think about is where in the body is the pathogen, right? So if a disease causes becomes septicemic and it's throughout the bloodstream, then the tooth roots are a great place to find the DNA because it's protected. You have blood supply, and so this is why for the Black Death and Justinian plague, your CENIEH pestis studies have all been pretty much DNA extracted from teeth. And teeth are great for DNA because you have the pulp chamber really protects the DNA. TB is not one of those diseases, right? And one of my graduate students actually spent a lot of time sampling different places on the lesions to see where we get the most DNA. And it turns out the closer you are to the lesion, and this makes osteologists unhappy, the closer you sample in the lesion, the better. And active lesions are better than healed lesions. So there's all these, she did a very careful study, which I think she's about to submit actually, looking at that. So we really have to think about where a pathogen is in the body and its likelihood of being preserved in the archaeological record. There is a big bias in studies in the archaeological record, and this is really hard to see, but there's a lot of white triangles in Europe. So most of our data is from Europe. It's cooler climate-wise. You know, there's basically, well, there is now a couple studies in Egypt, but very little data from Africa or parts of the tropics where we know infectious disease has had major impact on populations. So there is a preservation bias. There's also a bias in the sense of, you know, if a disease, if a pathogen does result in lesions, often it makes the bone more fragile. And so you also have issues there in terms of just preservation of the bone itself. So, okay, I've told you all the hard things. Here are all the things we hope to learn, though, when we can get the DNA. So understanding origins and relationships among pathogens over time and space. Obviously, we're interested in when pathogens first jumped into human populations, how they expand, what the evolutionary changes are. The Yersinia pestis work has been really fascinating because we found out that plague affected people as early as the late Neolithic, and we see changes in the pathogen over time that show that it adapted to being in a flea, and you know, there's questions about whether you could have certain types of plague early on, and that some of those developed later as you see changes in the pathogen, and we can actually see those over, you know, the last 6,000 years. We can also study the DNA of the host and ask questions about susceptibility, whether these big pandemics, you know, the Black Death is supposed to have killed a third to half of the population of Europe. So you would think there's going to be an impact. Those have been actually hard to find because you need a very large sample size to get at these small effects of many loci involved in immunity. We can also think about it just in the context of the individual and the population and the community, of thinking about how did communities respond to these epidemics in the past. So I'm going to talk about two pathogens. This is work that's going on in the lab. We are actually working on others, but we have nice stories from these two at the moment. The first of these is Hansen's disease, also known as leprosy, thought to be transmitted via the respiratory route. And this is a pathogen that most people think is not really with us anymore. But we've actually learned a number of interesting things about it recently. One is that there's actually two things that cause leprosy, the disease. One is Mycobacterium lepra, and the other is a closely related pathogen called M. lepromatosis. Where they came from and when they jumped into humans is still very much an open question, I think. As you'll see. So this is a disease that people think of as a very old disease. It's associated with significant stigma, and it's something that we tend to think of as not with us anymore. In the archaeological record, we see bony changes in the face. So M. lepra and M. lepromatosis like cooler parts of your body, so your nose, and your extremities, right, where your body temperature is lower. They cause nerve damage. They don't cause your fingers and toes to fall off. What happens is you don't feel your fingers and toes anymore, and you get cuts that become infected, and that is what causes gangrene and loss of digits. But you do see bony changes in the face, around the nose and palate in particular. It is a neglected tropical disease today. We see 200,000 cases per year that are new. It is treatable with a multi-drug therapy. And you'll notice, and you might be surprised, the United States has a darker color here. Probably Europeans, infected the armadillos. And so US-born people who show up with leprosy can usually remember having been in contact with an armadillo in the last 5 years, interestingly enough. So stay away from the armadillos if you visit. Yeah, so there's a lot of questions about M. lepra in humans, but not just in humans. I just mentioned armadillos. What about other animals? And I think when you study ancient pathogens, you very quickly become a One Health person, where you start to think about not just humans, because the story always also involves other animals. So a couple of other things about TB, how whether you have what's known as tuberculoid or paucibacillary leprosy versus lepromatous or multibacillary leprosy depends on your immune system. And there's different ways that it is described in the literature, but typically the word these days is paucibacillary, so few bacteria versus many. So the bacterial load, it's basically how well your immune system controls the infection. Ancient DNA has actually come in very handy because even at high bacterial load, pulling leprosy genomes out of a clinical sample is difficult. We actually use ancient DNA methods to do that. And so we build libraries and we pull it out. And so the study I'm going to describe involves samples from the 20th century and into the 21st that are from formalin-fixed, paraffin-embedded biopsy samples. So when somebody comes in and they have a little deadened patch of skin, they'll take a biopsy, they'll do the staining, they'll look for the bacteria, and then also use potentially genomic methods to pull it out and look for drug resistance as well. Interestingly enough, though, almost all the studies can really only get genomic data out of the higher bacillus load cases. We have a lot of trouble here, even with these very sensitive methods. So I mentioned that we also have to think about the animals involved, and over the last few years, my lab, as well as others, have been involved in looking at leprosy in other animals. And so cases have been described in chimpanzees, macaques, sooty mangabeys, squirrels, so there've been a lot of questions, and even around people's houses with Hansen's disease, it's been picked up in the environment. And so there's questions about, can it persist, and are there other vectors that could be involved? So in our study of primates that was published in 2018, we looked at 3 different primates. And what we found, interestingly enough, is that the animal cases are not basal. So this is a phylogeny. It's a family tree of leprosy genomes. With the little skull, that indicates ancient cases. But what we see with the primates is that their cases are, the pathogens are very closely related to pathogens in humans from the same region. And so we have to be mindful of that. So we don't see a clear origin of how leprosy, M. lepra, also I should say I'm focused on M. lepra. M. lepromatosis is very enigmatic. Most of the cases are in Mexico. And we know very little about it, other than both M. lepromatosis and M. lepra have a reduced genome, which means they look like obligate pathogens, and they separated about 13 million years ago. So it's been around for a while, probably not just in us. So another thing to think about there. But we also see from all the cases consistently a time to the most recent common ancestor that's about 4,000 to 5,000 years ago. Okay, so I said that. Now, one of the things you'll notice is that we have the deepest lineages are found in East Asia and in the Pacific. And so this got me very interested in what was going on in that part of the world. And so I began a project in collaboration with Keolu Fox and some others, trying to look at cases in the Pacific and learn more about that. There are a few pre-contact examples in the archaeological record. And we know, of course, that the Pacific was colonized, and remote Oceania was colonized by Polynesians, really in the last, depending on the part of Oceania, in the last 1,000 to 3,000 years, roughly. But then, of course, there's an extensive colonial history involving the US, Japan, British and French colonial powers in different parts of the Pacific. So there's a lot of question about where leprosy came from in the Pacific. So we began our study thinking specifically about the case in remote Oceania, and in Hawaiian, leprosy or Hansen's disease is the disease that separates families. But it's also been called the Chinese sickness. So there's questions about, was it introduced by colonial or trade in some way, shape, or form? And there are on the Hawaiian, or in Hawaii, and also Fiji and other places in the Pacific, there were leprosaria set aside, and that's a painful part of the history, the colonial history in Polynesia. So we began with samples from the Queen Charlotte Hospital in Hawaii, and the US has, obviously Hawaii is a state, but we have other territories in the Pacific, and all of the leprosy biopsies are sent to Queen Charlotte Hospital, or Queen's Hospital, and so we sampled 69 FFPE blocks ranging in age from about 5 or 10 years to 25 or 30 years old, from different parts of the Pacific. So, and you can see the stars here. So what did we find? We found that probably Hansen's disease arrived with the first Polynesians colonizing the Pacific. And we say this because of the dates, which I think I have, circled. Because of these branches, so in red are the ones that we sequence the genomes of. And when you look at them, they're in these deep branches, branch 0 and branch 5. Here's other East Asia, but these are the ones that are focused in Oceania. This one is from Marshall Islands that was previously done. Here's the macaque from the Philippines. And these dates go back pretty far when we estimate based on mutation rates. So this, and we see these radiations that date prior to the appearance of colonizers. So this has been very interesting. And so we've been trying to expand our sampling further, and we've done this recently in a way that we've actually also found a sample that is suggestive. It's in branch 1, which is related to some of the strains we see in Europe in particular. So that one potentially is one we thought, oh, maybe this shows evidence of colonization. We actually have looked more closely at that particular individual. So again, in our new samples, we have this branch 1, but it clusters with strains in the Philippines, and it turns out that patient had recently come from the Philippines to Hawaii. So we still have the story that's mostly a Pacific one. The rest of the new strains, when we expanded our sampling, are also still in these tight Pacific clades. And so we're now working to, And I had pictures of my graduate student and Keulu and various people involved, and those seem to have disappeared. But we are working to expand the study in collaboration also with folks in other parts of Polynesia to get a bigger picture and address questions about the larger picture of colonization, or the initial colonization of the Pacific, and whether the strains seem to follow what we know about how the Pacific was peopled. We also want to look at strains from before and after drug treatment was introduced to learn more about resistance, and also within patient variation, because we actually know very little about how leprosy is transmitted. So that's work that we're continuing. Yeah, all my people have disappeared. Interesting. Yes, many people involved. So that's work that's continued, that we're continuing and hope to expand in the next couple of years. Now for, more than 10 years, I've been working on TB. And as I mentioned initially, TB is part of a complex that some people call different species. Some people would say are different ecotypes. Some people would say would be subspecies. And I have to say that if you think species debates are bad in paleoanthropology, they're very bad when it comes to bacteria. So anyway, all of these, any of them, if you were infected by them, you would be diagnosed with tuberculosis and you would be treated in the same way. The same drugs would work. It's really only if you had drug resistance that they would have to switch out the drugs. So these affect a whole host of animals, as you can see here in the pictures that are showing. In humans, often, it's estimated that 1/3 of the world's population is infected with TB, but most of those people don't know it, right? Because most of the time, if you're infected, your immune system does a really good job of walling off the TB into a granuloma. And so it's only discovered if you have a chest x-ray and they see it. Or in the United States, where we don't vaccinate for TB because the vaccine's not very effective, you'll get the test where they put a little bit under your skin to see if you react. And then if you react, you would be treated. They would also do a chest x-ray. TB can remain latent for decades. As your immune system fails in old age, the disease can break out of jail. The pathogen can break out of jail and become active. If you become co-infected with something, HIV, for example, it'll break out of jail. The #1 cause of death in people who are HIV positive is not HIV, it's tuberculosis. Obviously, HIV is in there as well, but yeah. So it's really great at taking advantage of that environment of co-infection. It's called the wasting disease because one of the symptoms is you lose a lot of weight. If you live for a long time with TB, active TB, it can disseminate into the bones and cause even a kyphosis known as Pott's disease. So very much can cause some very severe cases, but this is in people who don't die quickly of TB, right? You have to live with TB for this to happen. But this is how we see it in the archaeological record, obviously, and we sample accordingly. Now, in the Americas, this was a big question because initially, through about the year 1998, everyone assumed that we got TB from cows. So, of course, we must have gotten TB during domestication and from being in close quarters with cows. But a couple things happened over the years both in the Americas and in the archaeology world and in the microbiology world. Jane Bijkstra and others described cases that looked very much like TB in the archaeological record in the Americas that are pre-contact. And of course, in the Americas pre-contact, you don't have cows. So there were hypotheses about did it come from, did people bring it with them when they colonized the Americas, or did they get it from bison, or, what happened? But we start getting the first phylogeny out of from microbiologists in 1998 and 2002. And what you can see from this is that compared to the human strains, the animal strains are more derived. They have all these different deletions that you don't see in the human strains. And so this pointed to cows not being involved, and actually suggests that we gave it to cows. Poor cows. After that, people really began, as the genomic revolution. Here's one of the things. When I was a graduate student, the population I was studying in the Americas for ancient DNA, there were cases of TB, and I thought, wow, this would be really cool to try to get at. Incredibly difficult with just PCR. But if I had pulled it out, all I could have said was, yes, TB complex versus no TB complex, in an individual that from the bioarchaeology clearly had TB, right? So it's not that informative. I would not have even had the genetic data to compare the ancient data to say something about its relationship to other strains. We don't get that until the early 2000s, actually. And that's when microbiologists start actually asking more questions along the lines of evolutionary biology. And here you can clearly see the animal strains are nested within the human strains from Africa. You can see these deeper lineages that are African and kind of rim of the Indian Ocean. And you can see that the strains spreading more rapidly today, that there are hypotheses that they are more virulent because of some genetic changes, arose more recently, possibly with increased population sizes. And so we've learned more about the distribution of these strains around the world, and that lets us really compare the ancient data. But how do you calibrate a phylogeny from modern data? Very difficult to do, and actually this is also where ancient DNA helps. Okay, so there's a very faint map of the Americas. Hopefully you can see it. So just a little bit about the archaeological record in the Americas. Not surprisingly, we see more cases of TB in places with higher population densities. This is true today with the disease, so it's not so surprising. Weirdly, the first documented cases were in South America as opposed to North America, which is a little later. And that was always attributed to preservation, right? Here along the coast of South America, it's very dry, amazing preservation. So maybe this is why we just happened to see these first. So our original hypothesis was that TB was brought in via the Bering Strait, and we should see relationships with those rim of the Indian Ocean strains. That is not what we found. This is what we found. Seals brought TB to the Americas. And when we got our results, we were like, that was not what we expected. And this is why those of you who are struggling with data that are not telling you what you want it to, it's often more interesting when you're wrong. It's far more interesting, actually, when you're wrong. We were wrong, and it actually took us then another year to gather comparative data because the TB strain that we had to compare it to was from a South American seal in a German zoo. So we had to start asking all these vets, do you have any cases of TB in South American seals, and can we have some? Right? And so we had to actually sequence the strains. Now, I should say that our samples came from this valley in the northern part of the Atacama Desert. And I need to talk faster, sorry. And we got 3 genome sequences with good coverage, so yay. And here you can see these ancient strains in Peruvians that are 1,000 to 1,200 years old, closely related to M. pinnipedi. Now, because we have the archaeological dates, we could then calibrate the mutation rate, and we figured out that TB in humans seem to be 3,000 to 5,000 years old. I would push that now to 6 based on what we've done since, but much more recent than people thought, because previous estimates ranged from 20,000 years to 3 million years, right? Big, big difference. Okay, so we hypothesize there's an African origin. I'm just sticking this dot here. We don't know. We have no idea the source. But then these deeper strains that I pointed out on that phylogeny earlier radiate into various parts of Africa and Eurasia. At some point, it jumps back into animals, right? We give it back. It's the gift that keeps on giving. And then we have the radiation later of these maybe more virulent strains. The animal strains, probably via rodents, jump into seals. There's the South African fur seal. I got to learn a lot about seals. And seals in the summer go down to various islands around Antarctica and on the coast of Antarctica, and that's where they all haul up. You see these pictures of seals, and they're all barking at each other's faces. It's great for respiratory disease. And likely this is how it spread from seal species to seal species until it affected South American seals and sea lions. And then likely spread up into, jumped back into humans and maybe spread further. So this led to a whole host of new questions, right? Is this just a zoonosis? Are other strains further inland caused by something else? Does the pathogen readapt to humans? Does it spread via human-to-human contact? And do other strains come from other routes into the Americas pre-contact? So this is what we've been focused on since the 2014 paper. And in 2022, we looked at strains from Bogota, Colombia, from 2 archaeological sites, one that sort of spans pre to post-contact, one that's definitely pre-contact. And this is at 2,000 meters, so there's no seals. Nobody's eating seal meat. We did nitrogen to check. But what we do find is they still cluster with the seal strains. So we do think that it becomes a human-to-human transmitted pathogen at that point. Since then, we're currently in the process of analyzing data from Aztec, Tenochtitlan, and Tlatelolco. So primarily from Tlatelolco. This is the Aztec capital and market city. It's estimated that 100,000 people per day, 65 to 100,000 people per day came to this market. So amazing. We also have additional data from northern Chile as well. And this is my very happy graduate student that she got 11 genomes. So here you can see what Diego Rivera thought it looked like would have looked like, but this is all under modern Mexico City and is no longer a lake. But interestingly, what we find is that these are seal strains, but they are actually deeper, older in a sense. The lineages are deeper compared to what we found in Peru. So this suggests that there were multiple jumps of this ancient seal TB into humans. And when we can use various methods of dating this using Bayesian statistics to try to get at when these events might have happened in time. And so that's something that we're actually currently refining, but these dates are probably in the ballpark. But what this suggests when we look at it more closely is that there was a jump somewhere in South America, and we don't know where, but we do know that there were trade routes between northern Ecuador and West Mexico, and likely that may have been a way that those strains reached up into Mexico. Later, new strains would have been introduced, and that's what we're picking up in Peru. About 1,000 years ago. And one of the cool things that we're trying to do now is in places where we have enough genomes, really try to do a genomic epidemiology, much like they've done with SARS-CoV-2 strains. And so that's something that's in the works. So what we see is both evidence of something that looks like an outbreak, as well as some diversity in strains in Tlatelolco and Tenochtitlan that suggest either that strains were being reintroduced because of the market, or that there were some outbreaks, but things that lingered for a while as well. And both of these things actually could be true. We've also looked at samples that are post-contact, and what's really interesting there is we see a replacement. And when you look at those strains more closely, what you find is that the strains are European. They're actually part of this lineage 4, some of which you can't really see here, but trust me on this. But what's interesting is that they sort of match what we see in Europe or East Asia of where the likely first colonizers are from, right? So for Mexico and New England, they look more like European lineages, but interestingly for the Alaskan strains, they look more like strains that are found in parts of eastern Russia and China. And of course, Russian fur traders were the first to contact. Now, however, Inuit and Aleut also had extensive trade networks over the Bering Strait. So it's a little hard to tell there, but very cool. So this is something that we're following up. We're also looking at the human genomes to try to look at the effects of this new pathogen being introduced. And what this has been done in Europe with the introduction of these L4 strains. We see some changes in the genome that are suggestive of people who are more susceptible with alleles that cause more susceptibility. Those allele frequencies go down through time. And some similar work has been hinted at in the Americas. We see all these genes that have been linked to tuberculosis susceptibility and resistance showing up on selection scans in South American populations. So, and there's been some dating of this that suggests this signal of selection starts in the last 3,000 years, which is also quite interesting. So something that seems to match up with some of our estimates of potential jumps. So I want to end with a couple things to think about that pushes this back further in time. I've talked to you about pathogens that jumped into humans probably in the last 5,000 to 6,000 years. This is when human population density increases. And George Armelagos, many years ago, came up with this idea that in humans we have, sort of heirloom pathogens and souvenir pathogens. And so I've been talking to you about souvenirs, really, of the Neolithic Revolution. But, what would have affected people earlier? And there are hypotheses and I think some clear evidence of things that likely did, whereas tuberculosis and others are more density dependent. We need high population levels for them to spread. And so, so it's lately been very much a story in ancient pathogen DNA of the effects of the Neolithic Revolution and the increase in population density. But I think people are also thinking about, pushing that back further, and there's ways that people already have. These are the things that likely affected humans given our low population density through most of our evolutionary history. And we don't see a ton of disease in the record. I mean, we see chronic diseases, arthritis and things like that. But obviously only some diseases cause change in bone. And we also don't see signs of the biggest killer of kids, right? Biggest killer of kids, diarrhea. Nobody talks about diarrhea because you don't think about it, right? It is the #1 killer of kids. So pathogens, but there's very few studies of kids in the archaeological record that have looked at ancient DNA of pathogens. So that's another aspect. We do have data from some sources. So the microbiome, dental calculus actually preserves DNA really well. And you can see some different colors in here, but the red ones have been linked to periodontal disease. And so you can see included in this sort of bit of color, and you probably can't read it, but these light color, those are Neanderthals. So we do have microbiome data from Neanderthals. And we can see the similarities with modern humans and some differences. I've only shown you part of this, but there are some similarities more with our closest relatives. We can also use the genomes that we have of archaic humans to make inferences about pathogen pressures. And so people have done that as well. And that often takes advantage of a lot of the genomic studies of clinical samples, clinical patients also. Oh, I didn't realize that. Okay, that should have come up first. Anyway, so there are ways that we can kind of get at this through our longer evolutionary history, and hopefully we'll get more data in the future about this as well. And I end by thanking all the members of my lab and the First Nations and Indigenous communities that have given us permission to sample their ancestors. I also thank all those who have given us funding. And here we are in our new building with our new ancient DNA lab, which makes us very happy. And I also have many, many collaborators that I've had the privilege of working with as well. So thank you.
Wonderful. Thank you so much. I know I have a lot of questions. I'm guessing others have questions.
I think she's waving a microphone.
Oh, microphone. Sorry. Great. So the people online can hear us better.
Yeah, so that the people online can hear as well. Good idea. Hello, if you're online, I'm glad you're with us. So we have, I'm sure there's questions. I know I have lots of questions, but maybe people in the audience. I don't know, Carla, can we get questions from people online? Okay, yeah, so if you're online, please enter in, where would they put a question?
In the chat?
In the chat? Okay, in the chat. Yeah, and we'll be looking. So,
Okay.
Shall I go first? Okay. Oh, actually, way in the back. Okay, I'm coming. Carlos, yes.
Thank you so much. Wonderful talk. I have a question. How do you, you mentioned that you measure, well, not measure, but you balance the mutation rates for calculating or to locate the previous strains or previous ancestors of these bacterium. How do you do that? How?
Right.
Well, I guess there are. I mean, not in detail, like, but.
Yeah. So, so if you think about this from looking at ancestors of different mammals, right? So normally people calibrate the molecular clock based on fossil data, but we don't really have fossils for bacteria and viruses. We have some, like we know that the oxygen, the Earth became oxygenated about 3.5 billion years ago, or 2.5 billion years ago, which is a terrible calibration point because it's so old. And then for TB, people first, threw TB strains in the freezer about 100 years ago, right? So we have a really recent calibration date and a super ancient one. And that's really terrible for trying to figure out a mutation rate. So the archaeological record is actually super helpful. So when we published the 3 genomes, we knew from the archaeological data, C14 dating, but also just from the cultural material, we had very good dates for when these individuals lived and died. And so using that, we can then say, okay, if that strain is related to this other strain that we sequenced last week, right? We know the amount of time that separates those 2 lineages, and then we can average out what the rate is, and then we can infer that over the tree. But with the statistical methods we use, so we had 3 different dates, it would think about that. As we've gotten more data, we can add those dates in, and then Bayesian approximation helps us figure out, what the average mutation rate is. It also lets us, because I mentioned that sometimes bacteria and viruses will suddenly speed up or slow down, we can also factor that into our estimates and look at, when we have enough data, we can look at variation on different lineages to see how that impacts our estimates of that most recent common ancestor.
And you also mentioned that, bacteria are quite promiscuous, depending
Yes.
on the environment that they live, etc. But could you infer about bacteria communities in certain areas? You mentioned that you basically had to, when you're sampling, removing the environment, etc. But could you, from the data that you're receiving and that you're getting from these DNA extractions and analysis, could you also think about the communities or DNA that might be
There too.
crossing over between?
Yeah, no, that's a great question. So we're really, I forgot to mention this, so this is great. We're really lucky that Mycobacterium tuberculosis and Mycobacterium lepra are clonal. They don't recombine often, really much at all, to be honest. So we don't see evidence for recombination. We can do various tests to make sure that is the case. So that's super helpful. They also don't have plasmids, so we don't have to worry about that. What we do find, though, is sometimes someone will be infected with more than one strain, and that's complicated to pull apart the two strains, the data. That happened with the case from a mummy from a crypt in Hungary that's about 500 years old, I think. And so bioinformatically pulling those two strains was very difficult. When we're analyzing the data, and what we will see sometimes is it looks like there's, instead of having what should be a homozygous sequence, right, everything because it's clonal, we'll see variation that, in other words, it looks like it's a little heterozygous. Some of that could be just that given how long somebody's lived with the strain, there's kind of, it's developed mutations in various parts of the body. But some of that is the signal of the environmental mycobacteria. And so we have to work hard to pull apart what's likely the environmental mycobacteria from what's the pathogen. And so there's a lot of thinking about how to adjust parameters to try to get an accurate sequence. And sometimes that's difficult, particularly if we don't have a high coverage genome. The noise can be quite large.
Thank you very much.
Do we have another question? Okay, then I get to ask mine while I walk back. So I remember, watching in the early days the concern with all the contamination of other human DNA. So it's interesting then to see how you are working on all of these different diseases, these different, so from viruses, bacteria, and with really recent human populations. So it's how much do you still have to worry about contamination and cross-contamination, and that, how complicated is it now, or are with all of the modern technology that you don't worry as much about all of the cross-contamination, working with all these different people and different infectious agents?
We still worry about it. You saw the little suits that my grad students get and postdocs get to wear. So the biggest concern is us. And so that becomes, hopefully I seem to have a healthy lab group, so that's good. So the ancient lab's a completely different building from the modern lab. So any control sequences or that all happens in the modern lab in a different building if we're doing something modern. We tend not to work on the modern things. Formalin, fixed paraffin embedded samples look just like ancient DNA ones because of the process of formalin is terrible for DNA, right? And so it really damages it in some of the similar ways. So typically the DNA fragments are 60, 70 base pairs in length for those samples. So we do analyze those in the ancient lab because they act that way, shall we say. So we're very careful about keeping things separate. We also think a lot about, environmental sources. Sometimes I'm thinking about people and their cats and dogs and what they might carry. There was a surprise that squirrels in England carry both M. lepra and M. lepramatosis. So there's a lot we don't know about where these pathogens are in wildlife in particular. But we do keep the wildlife out of the lab. You cannot bring in your pet squirrel or your pet dog. So, So we are always concerned about that. We do take advantage, actually, of these patterns of damage to help us authenticate things. And we always ask ourselves if the results are making phylogenetic sense.
Thank you so much. Any other questions or Carla, anything on that? No, well, sorry, one more.
Just a quick question.
Oh, microphone. Oh, sorry.
Sorry, you said that TB is not very good. We know we get the pathogens from TB, but TB is not the case. From these samples from Mexico, did you sample teeth or did you went for?
Oh, yeah, great question. So we've never gotten TB out of teeth. The reason we know so much about Yersinia pestis is that people are sampling boatloads of Europeans and mostly their teeth, and they'll be actually asking questions about the human DNA, but in the process of extracting all the DNA, they'll run it through a pipeline to see if they pick up any pathogens, and then that's how the late Neolithic Yersinia pestis was discovered. It was like, hey, look at this. That's cool, let's capture it, right? And then they did the capture. But even though we know that TB was a major cause of death for centuries in Europe, we never see it in the teeth, right? So this is why, my graduate student was really kind of sampling around the lesion to figure out where's the best place to sample. And it is the active lesions around TB. Now, that means that a lot of times if somebody died very quickly from TB and their bones don't show any evidence of TB, you don't see it, right? We now have one case where it looks like we see it in the teeth. And my hypothesis there, and so I've purposefully sampled some kids. So if you look at how tuberculosis disseminates in kids, it's different from in adults. So you often will see it in epiphyses, epiphyseal regions, because they're growing, right? And so it disseminates in a different way. And so my thought was, well, maybe we would see it in the teeth of kids. So we have one or two maybes that we're in the process of working on, but then capturing the full genome is where we're at, and we have some low coverage genomes. We're now going back and trying to get more, because we're trying in that same valley in Peru where we have 4 genomes to expand that to get a really denser sequencing. The most cases of TB in the archaeological record in the Americas is this valley in Peru. And so what we'd like to do really is do a kind of a genomic epidemiological study of that valley. So that's what we're trying to work on there. And so hopefully I will have more answers soon.
Well, I was asking because, I'm sampling a lot of
Yeah.
material in Mexico, but I'm focusing on teeth and
Yeah.
bone, and because we're not really looking for teeth. But
Yeah.
I was just wondering, maybe
Your
if I have
odds,
some too.
yeah.
Some TB.
I'll be surprised if you see it. Possibly one or two, more likely if they're younger, is going to be my guess. But yeah. Yeah.
I'm only working on teeth.
Yeah. But, you know, the other thing about the European record, it's almost always adults and almost always teeth. And I think that's why you don't see it.
Thank you.
And there is a question from online.
Okay.
Where do you get your samples from for the microbiome work?
Right. So we are studying the microbiome in northern Chile. I have a graduate student, Mario Pata, and he's interested in how the microbiome changes from archaic populations who are marine, taking advantage of marine resources, to when you switch to agriculture in the history of the Camarones Valley. And so there, what you do is, when you go to the dentist and they scrape off that stuff, that's dental calculus. And so you see it in the archaeological record and you scrape it off the teeth. So don't clean the teeth which for years people did, and now we're like, ooh, there's data. So you scrape off a little bit of that dental calculus, and then you use very similar methods. To the extraction is slightly different, but then you use a shotgun sequencing to look at all the DNA there. And that's really a case where you're looking at the microbial community and looking at which part of that community might be involved in tooth decay and which part might be, it turns out a lot of your bacteria have handy jobs. They'll convert different nutrients for you. So we see those signatures as well in the dental calculus. And sometimes you can also pick out the DNA from the food, plant or animal DNA. So there's actually quite a lot you can learn from dental calculus. It's pretty cool.
So we should probably wrap up. Or actually, one more question? No, okay. Yeah, we should probably wrap up. We've taken up a lot of your time. So thank you so much, and please everyone
Welcome.
help me thank Anne.
You're welcome.